been asked to talk on therapeutic drug monitoring of antifungals, mainly the principle and performing the assay in resource limited setting. So first of all, you had to know that in a group of, when a patient group is there, so you with the same diagnosis and with the same prescription, but whenever you give any drug, it does not behave in a similar way in every person. For example, you can see that in case of, so it may be drug may not be toxic, but it may be the beneficial and curing the infection or other thing. Drug could be a toxic but not beneficial. This population is very, very less. And drug is toxic but it is beneficial. Again, this is very, very less number of patients. And also you have where the drug is not toxic as well as not beneficial. Not beneficial. So in this context, we need to assess that which particular patient is getting beneficial or which person may get a toxicity due to the drug. So for this, there is what we can do is TDM is the one which is which helps or guides the clinician to predict efficacy as well as to see that say that whether a patient may go into toxicity and other things. So therapeutic drug monitoring is not only done for antifungals. Antifungals, though it has been from last 2 decades, we are doing it for voriconazole. So mainly applied for many of the toxic drugs that is their treatment for other diseases. So in definition, it is therapeutic drug monitoring is the use of drug concentration measurements in the body fluids as an aid to manage for the management of the drug therapy for cure, alleviation or prevention of the disease. Generally speaking, with regard to antifungals, so we need to do TDM mainly to see whether the dose is achieved exactly properly what is required to be efficacious, or the dose is more so that the patient may turn into the toxicity. So here, what you should understand the therapeutic range. So if you give any drug to a patient, so I try to assess the concentration of the drug within the plasma. So you have a therapeutic range. So therapeutic range is the one which is a dose in such a way that the proper or expected response is seen or achieved. So if the range therapeutic that dosage is lesser, or if the range is lesser, then it will be a subtherapeutic and it will not be useful and cure the infection. If it is more, again, you will have a toxicity. Therapeutic index is the one which gives you very, very good clue. So blue line is the one it is which what is expected or therapeutic effect. So you can see the graph like this. Whereas if you go on increasing the dose, then you can see the graph like this, where it is a toxic effect. The distance between the effect and the toxic effect, TC50 or ED50, TD50 we call it. So this one is called as, this is very, very important to understand. So for example, some drugs will have a very large therapeutic index. So you can play with the dose, you can little bit reduce or little bit increase depending upon the dose that has been, plasma concentration that is there. So, but for some drugs, you have very, very narrow therapeutic effect. This one index. So in this particular situation, especially, you need to have a therapeutic drug monitoring to assess whether the dose is efficacious or it is toxic. So for few antifungals only, that's why we do not have there is no need to do this one TDM for all the antifungals that is there. So I'll come to it later in my presentation. So when to do the TDM? So as I told you, when you have a narrow therapeutic index, when there is a direct relationship between the drug and the plasma and toxic effects. So when it is known that above this particular concentration, you have toxic effects, then you need to do a therapeutic drug monitoring. And in many of the situations, we know that in an fungal infection, deep fungal infection, we do not have any particular clinical sign to say that the patient is improving or not. So it will take longer time. So at that time, whether you have to increase the drug, decrease the drug to have a good efficacious, you don't know. So in those situations, again, you need to have a therapeutic drug monitoring. And also you need to, when there is a variability in the steady state of the plasma concentration is known for that particular drug, then you need to do. And also above all, you need to have a method for doing a therapeutic drug monitoring for that particular drug in question, which you want to test it. So major indications is purely defined clinical endpoint and non-compliance, especially in case of chronic diseases, we will prescribe antifungal for a longer time. So at that time, patient may have recurrence or this one. So to see whether the really it is a compliance is there or not, there you require. And therapeutic failure again, and then wide variation in the metabolism of the particular drug and major organ failure, and also to prevent the adverse effects. So drug concentrations cannot be predicted from the dose itself. So whatever dose you give, you cannot say that, okay, that same concentration is achieved in their serum or at the place where the infection is there. So you need to understand about the PK/PD factors. So I will not go into the details of the PK/PD factors. So just one slide, just I'm showing you on PK/PD. So ADME is an absorption, distribution, metabolism, and excretion. So this is the basic principle. Concept is when you give any prescribed any particular drug, there is an adherence, and then it will go into the blood and distribute into the different tissues, including the site where it is required, and then it is excreted. So all this function is called as pharmacokinetics, and this is definitely variable. When the pharmacodynamic is, again, you have to see that where exactly we read a response, that is at the site where we expect a response, whether effect is there or not. So that is study called pharmacodynamics. So I will not go into that thing. So just this is the curve, you know about it already. So what you see is here, whenever the drug is given, so the concentration increases based upon the time. So every drug will have an Cmax, that is highest concentration at particular time, it will reach an highest concentration. And then after some time, it will come to lowest concentration. So again, you need to have an off-life. So off-life anywhere in between the highest concentration and the lower concentration will be your therapeutic index. So you need to identify that particular thing. So that's why for that pharmacokinetic and pharmacodynamic studies have been done. And for all the antifungals and for most of the most common diseases, we already know these particular values. So we need to just do a therapeutic drug monitoring and see whether it is efficacious or not. So when you are going to do a therapeutic drug index, you need to take a trough concentration. So trough concentration is just that plasma or even serum, you can take it. That should be drawn at the trough. Trough means just before you are giving the next dose. So for example, you are giving a dose in BD. So tomorrow morning, the next day, he has to take a next dose. So just before taking the next dose, you need to take the samples in order to do that TDM monitoring. So this is CSS minimum, we call minimum steady state concentration. So at that particular level, so minimum steady state will be there before taking the next concentration. So that's why at that particular time you have to take the samples for doing TDM. Peak concentration, this is the time after the dose at which the peak concentration is achieved. So coming to the antifungal TDM. So when you do the antifungal TDM, so for a different the context you have. So for example, pharmacokinetic variability. What you see in case of children, pharmacokinetics is different. Neonates, it is different. Elderly, obese, the mass is too much. The same dose you cannot give in a 50 kg and the same dose along for an 100 kg person. So organ dysfunction, absorption, and other thing variation is there. Critical illness such as hemodialysis, hemofiltration, extracorporeal membrane oxygenation, cardiopulmonary bypass. These are all the major things where we consider for doing a TDM in case of antifungals. Again, you need to do and changing pharmacokinetics. For example, physiological instability, critical illness, diarrhea in those situations. Interacting drugs when the patient is taking. So some drugs will interfere in the absorption. Some will make absorption excess. So when you know that the interaction drugs is there at that time, compliance I already told, when the patient is in poor prognostic. So these are all the main certain indications for doing an antifungal TDM. So when it is generally indicated for an mold active triazoles, itraconazole, voriconazole, posaconazole, and flucytosine. So for fluconazole, we do not have much data. We have a data, but all the evidence suggests that there is no need to do an TDM for fluconazole. So it is especially when you are giving a fluconazole in critically ill patients who is on hemofiltration, at that time you may have to do it because of the loss of the fluconazole during this procedure. And for echinocandins, again, there is no, the data, all the data suggests that there is no need to do a routine TDM for echinocandins. So this is a table that has been compiled by Ruta B. So which is published. So till now it holds good. But for a little bit more data are there for posaconazole. So itraconazole, so for example, you can give TDM when you are giving the drug as a prophylaxis or when you are giving the drug for a proper treatment, as well as when there is toxicity. So you have to do for itraconazole, not for toxicity. The evidence is recommendation is very weak. So you have a strong recommendation both for prophylaxis and treatment when you are giving. For voriconazole, you have to do for the treatment purpose as well as for the to look for the when expect for the toxicity. For posaconazole, mainly it is given as a prophylaxis, especially in this hematological stem cell transplant and other patients. So again, when there is a strong recommendation for doing an posaconazole TDM for when it is given for prophylaxis as well as for the treatment. For fluconazole, they all are against. For echinocandins, again, all are against. For polyenes, again, amphotericin B and glycopulvin, there is no need to do a TDM. So the recommendation is strongly against. So, but flucytosine, again, only for toxicity purpose, you have a good evidence and that a strong recommendation has been done. How to measure? So coming to the bioassay mainly, so you have different methods. So bioassay is a simple method. It is just like performing an antifungal susceptibility testing or disk diffusion, or for bacterial disk diffusion, how you do. So by just following this procedure with the standard quality control, you can do a bioassay. So, but the gold standard for doing a TDM is by doing it with an HPLC. This technology, but the only thing is the problem with HPLC is if you have a machine, so that technically you should have an experienced person. If you have a machine, so you can do it within 2 hours or so, you can come and do the things, that will be easy. So, but especially everywhere you cannot, in the research lab like us and in AIIMS and other places, you will have an HPLC and you can do it very easily. But even liquid chromatography is another technique that is there. Again, this is cumbersome and you need to, combining with the mass spectrometry. So you need to have only in big research labs, you have these kind of instruments and it is very difficult. So if you can, I can tell you that bioassay is one of a very important thing. And I'll come to the results. We also performed the bias. We used to do bioassay about 10 years back before getting this HPLC machine ourselves. So in bioassay, as I told you, it is just like doing an antifungal susceptibility testing by disk diffusion testing or antibiotic susceptibility testing. So when you want to do an bioassay, always you should not take the tablet. You should always buy this one pure form of the drug should be there. So any antifungals you are doing the bioassay, it should be in a pure form. So the medias actually they have evaluated many medias. For example, when they are testing for itraconazole, when they are testing for voriconazole, when they are testing for posaconazole, different medias have been used. But bioassay is a little bit if you use an quality controls together. So if it is giving a good results, then you can use, for example, we were doing an voriconazole TDM 10 years back. So at that time we used to do with the RPM agar. So other evidence also is there. We also have used this peptone agar for doing this one. So RPMI is recently very good. It has worked very well with our hand, and also others also have used this particular media. And only thing is you need to add an within the standard RPMI 1640, what we get it for an broad dilution method. So for that you need to add an agar and then 2% glucose. So this is the only thing that is required. And coming to a strain, so as I said, so when you are doing a bioassay, you are looking that you are putting particular amount of concentration of your antifungals, and then you are testing that how much inhibition is there against a particular strain. So this strain, when we are using, should have particular characters. For example, Candida kefir, this is the one standard strain that has been used very largely because quality control and all the other precisions, accuracy and other things have been looked when they are used for particular antifungal. So that's why we also got this particular, we have this Candida kefir. So we can share this particular strain with you all. This again an 80cc strain. So it's also called as San Antonio strain. David Denning used to use this long back, 20 years back. We used to do it for itraconazole. So that's why we got it from him, and then we are using this strain. So this particular strain is, it is, for example, it is sensitive to itraconazole. When you are testing any drug for an TDM, so it should be sensitive to that, but that strain should be sensitive to a particular drug. So that is one of the important character. If you want to use any other strains, you can use your own strains also, but you should follow all the quality control, this one. So I will come to it, all the quality control. When it is giving a good results, then you can use it your own strain as a standard strain. So when you do the bioassay, if you also have to compare it with a standard gold standard HPLC, if you want to select your own strains. But this particular strain is there. If anybody wants to perform an bioassay, you can, it is welcome. You can write to us and you can take this particular strain for daily. And then you need to have a sterile normal humans pooled serum. So this is again for a control purpose you need to have, because you want to look that there is there any other component in your serum sample or in your procedure which is inhibiting the inhibiting the growth of a fungi. So that, and you need to use a bigger plates. So because you are testing it as a different concentrations and different samples. So if you use an big plates, then that will be better. Even plastic tray can be used for doing it. And you need to have a vernier caliper for measuring the zone of inhibition. So standards, when you have taken antifungals, you should have in standards, because you need to have a graph, standard graph. We have to first graph it out. So that's the one thing that is required. So you will take an, for example, voriconazole if you are using. So voriconazole at different power 1 microgram per ml, 2 microgram, 4 microgram, 8 microgram, 16 microgram, you take it. And then you see the zone of inhibition one side and concentration on the other side, and plot a graph. So internal controls, you should use it. So for example, already known sample of this much concentration which is there, that also should be used. And then 100 microliter RPMA agar and mix it with 5 ml of inoculum containing 10 to the power of 7 cells. So this one actually varies depending upon the strains which you are using, or if you are standardized your own this one. Inoculum standardization is again a very, very important one. That's why we follow on standard methods. If Candida kefir, already we know that this much particular cells, it will give this much zone for an voriconazole against voriconazole. So if your results are giving this much, and you can use this one. And then what you have to do is mix well, pour into the plates and allow this particular medium to solidify for 30 minutes. And well, you have to cut. You have to well cut. You should have a template to cut the well. Otherwise, you can use this pasture pipette, back of the pasture pipette or something which is small tubes so that you can where you can add the samples. That is this is the particular this one which can be used if you have. Otherwise, pasture prepared you can do it and to add a sample. So that drug should be allowed to pre-diffuse at the room temperature for 30 minutes. So once you had a standard controls which you have prepared, 1 microgram within the thing, so you put it into a well and then leave it for some time for 30 minutes and incubate at 37 degree for a minimum of 18 hours. So here I did tell you that whatever culture which is there, so the culture inoculum you prepare it and you have to add that culture to a media. So here the media along with the culture at 50 degree, you cool the culture and then you have the media and then your plate is ready. Then you punch the wells and then in that you can add a different sample. So and then later for at least 18 hours you have to leave it and then you can measure the zone of inhibition and multiple people should read it and then plot the log10 concentration on y-axis and zone diameter of the x-axis and concentrate depending upon your internal controls and sample using a straight line equation you can draw. So this is how it appears when you do a bioassay procedure. So you can see that these are all the standards 8 microgram, 1.6 microgram. This was the earlier one which we have done. So 3.2 microgram, 6.4 microgram, 20 microgram. So based upon the zone diameter as well as an concentration, just you plot the graph and see if you get a straight line, your standard is really working. So you should get a good graph like this when you plot it. And then corresponding value from your sample, patient 1 or patient 2. So you are going to plot it somewhere you will get it here. So depending on that, you will have a concentration of this one. So it is a very simple thing and very easy thing. Just you need to have a controls. So one serum control is very, very important. And then the drug control, concentration control is very, very important. So evaluation of HPLC. So that's what when we did it for protein serum sample, we evaluated, and then we also do then HPLC. So many publications have been there. So these are the results. At that time, we used to get a sample. Even now, we get it from many places for doing an TDM for. Usually now we do it HPLC. So we have done it and you can see here the results of bioassay as well as HPLC. So it is almost very, very, very close and this is mainly for an voriconazole. So for voriconazole, really it works very, very well and anybody can start with a good quality control or you have to have this external quality, participate in external quality assurance program. So I think Dr. Karuna can take an initiative. So at least we also can, because people are not doing, we are not sending the sample for this one TDM. So we can in future, if many people start doing this, so definitely we can incorporate that in our EQS program also. So and that in particular, this one we found that correlation between the HPLC and it's 0.9994. So it is a really very, very good. If you use these particular medias, that is RPMI and the methods that I told you, and then bioassay method is a good reproducible and correlated well with an HPLC method. And when the dose of voriconazole was increased in 2 patients based upon our report of from a bioassay, reduced in 3 patients and 5 patients were found in optimum drug concentration for improved clinical outcome. Again, you can see that here. Again, this paper is long back in 2013 they are done. So we actually did not publish this one, but only for 8 patients they have looked and they have published and see the precision accuracy compared to quality control. It is really very, very good and comparable to this one, especially for voriconazole and even for posaconazole. We do not have any published data, but just we need to again determine. But it's very early to say that it's very good. But posaconazole only 1 publication there was not much inference whether bioassay is really gives a good value or not. So bioassay suitable for samples with antifungal monotherapy. So always this is a very, very important thing. If the patient is a combination of the antifungal therapy, you know that one antifungal, if it is there in the serum, then it is inhibiting. So you can find out the inhibition of only that particular thing, antifungal. If the patient is on voriconazole and caspofungin also will have an added combination, will inhibition, so that you will not get a correct voriconazole levels because the zone of diameter will be more because of the effect of a caspofungin. So bioassay in the case of HPLC, because the peak will be given at different different places in the graph, so it can clearly differentiate caspofungin level as well as this one. So this is the main drawback of this one bioassay. But if the patient is on monotherapy with an antifungal, then definitely it can be useful. Combination of antifungals, again, I already told that combination will not be useful, but still, one paper they have used this Candida parapsilosis, which was sensitive to voriconazole, but resistant to this one caspofungin. So patient was having a combination. They had around 4 or 5 patients given combination. In that case, they standardized this Candida parapsilosis isolates to use it as a standard, and then they have identified. But that when the combination, we need to work on it. We should have such kind of different combination of this one isolates and QCs to have a good this one. Results of itraconazole is generally very high. I will come to it later, because itraconazole you will see it in 2 different forms. So that's why you will get a very high, and it's very difficult for bioassay. So HPLC is always better. So as already I have told you about quality control. Almost it is very, very important. You have to participate in a quality. So certain recommendations, for example, voriconazole TDM recommendation. Generally, you do it for this and you know all the this one where voriconazole, especially it is given for mainly for aspergillosis and many other infections. Its pharmacokinetic is non-linear, and dosage adjustment is necessary. So because of the non-linear pharmacokinetics, due to an unpredictable changes in the drug exposure, oral bioavailability may be lower in children. Again, there you need to do an TDM, and you know that this particular thing is metabolized by the liver through P450 enzymes, which is mentioned here. And in voriconazole, one consideration is, you know that if there is people who have a polymorphism in this CYP2C19, then absorption is really very, very less. So that's why it is now, it is a well-known fact. So in India, we don't know how many percentage of people are having this particular polymorphism in this particular thing. So this association is very, very this one. So that's why, so at that time, if you do an TDM, so just look for this very polymorphism or not, that to those patients require a higher dose of an voriconazole, because it gets excreted very fast. So in voriconazole, as I told, both it can be used for prophylaxis as well as for the treatment. So TDM value should be more than 1 milligram per liter. So the trough value, whatever you are determining, so then you can have a good this one. So maximum limit is about more than 6. So you consider it as a toxic. So then you need to reduce the doses. And for treatment, again, measurement of serum trough concentration should be done. So always when you do when you are doing a TDM, you should know that when is the sample, when you can try to see. So if you give a dose for caspofungin, actually steady level, it will take for about, not for caspofungin. So for any antifungal, right, to reach, it will take at least 2 to 3 days. If you are giving an oral formulation, IV formulation, okay, you will get the levels very fast. So during those situations, so here actually what they say is, so trough concentration should be done within 7 days of initiation of the therapy or following the dose adjustment. And you should do it when there is an interacting drug, when you are adding it or when you are stopping it. and also any absorption issues and other thing. And when you see any particular toxicity, if it is there, like eye toxicity as well as liver toxicity and other, which has been seen with the voriconazole. So for itraconazole, again, you have many issues with itraconazole because mainly you know that it has been given for many fungal infection. So oral candidiasis, pheohypomycosis. If for invasive aspergillosis, if it is refractory or intolerant to voriconazole, then they will give. But now we have both posaconazole, even anidulafungin, this one isobuconazole is there, as well as this caspofungin is there as a second line of the drug. So itraconazole is not much preferred as a second line in the refractory cases, but still for chronic aspergillosis, cavitary aspergillosis, CTA cases, allergic fungal disease, dermatophytosis, and sporotrichosis or pheohypomycosis, there it is become it is preferred. So we should have a method for this one, but unfortunately you have many issues with an bioavailability of this one because of non-linear pharmacokinetics, and the formulation is very important. For example, if you have a capsule formulation, then bioavailability is really very less. If you have a solution, but solution is rarely available in India, so it has got an higher 30% higher bioavailability, and also it does not interfere much with an pH this one. So TDM generally here what I would like to say is generates a multitude of metabolites including hydroxytraconazole. So the discordance in an bioassay is mainly because of this particular thing. So you will measure both the itraconazole level as well as hydroxytraconazole level. So you cannot differentiate which activity is because of which one. So itraconazole level, main itraconazole level is the one which is very important. So because of that, bioassay may not be useful for this one. It will generally give about 5-fold higher value compared to the HPLC or the mass spectrometry. So that's why bioassay may not be of much useful for this particular situation. Again, precipitation of itraconazole standards occurs in the bioassay due to the poor solubility of this itraconazole, and so that also will give you an overestimation of the drug concentration. So itraconazole again, so the drug concentration should be more than 5 microgram per ml, and it is again you could do it after the 5 to 7 days after the initiation of the therapy or the dose adjustment, when you give both for a prophylaxis as well as when you use it for a treatment. Posaconazole, now it has been generally known that it's used as a salvage therapy for aspergillosis, and even in mucormycosis, FDA has approved for an as a first line therapy for mucormycosis. Along with this one, amphotericin B. So, and for coccidio, chromoblasto, and all, it has been used as a salvage therapy. So it is again, this one will inhibit an CYP34 activity, and there is a significant absorption issue. That's not much. Increased oral bioavailability with the food, increased absorption with an acid environment. Fat actually will help in absorption of this one, but there's no data that suggests any correlation between toxicity and the drug exposure. So you should always see that for prophylaxis, it is 0.7 microgram it should achieve steady state, and then for treatment purpose, after 48 hours of initiation, you will generally do this one posaconazole prophylaxis level stadium, and for treatment purpose, it should be more than 1 microgram, and within 7 days of the initiation of therapy, you should start doing the thing. For flucytosine, again, you all know that it is generally not given as a single agent. Mostly it is given as a combination. So again, combination here, that's what it's a bioassay if we do for these patients, especially in cryptococcosis when they are giving. So then the flucytosine level can be not be achieved. So we need to have an amphotericin B resistant isolates in order to, but till now we do not have but such particular isolate to test an flucytosine when it is given with the combination with the amphotericin B. So that is a major difficulty for bioassay. but HPLC can be used it can be easily being used, which we are doing it for all these particular antifungals. We are regularly doing the this one HPLC method of TDM. So again, these are the issues. I am not going into it in detail. So mainly for flu cytosine, when there is an this one toxicity, we look for that purpose. TDM has been done, and trough concentration should be 20 to 40 milligram per liter, and within 72 hours of initiation or this one, we should collect the samples for doing it. So thank you very much for your kind attention. I will be happy to have an proper discussion on this topic if there is any questions and other. Thank you very much for your kind attention.